hypothesis that every bacterial cell, they were studying E. coli, that the E. coli had a system where the foreign DNA in the form of the phage DNA in strain B was actually being chopped off. Whereas in strain A, since the phages survived, there was no chopping off and no cutting of DNA. And the second time, when you introduce those one or two plaques that came in the first experiment on B strain, and if you put it back again in the B strain, you got a lot of plaques, which clearly suggested that once the phage gets into the E. coli cell, which is the host, there is not only restriction, but there is some kind of modification that happens so that the second time when you introduce, the cutting doesn't happen at all. And this was the basis of the phenomena called restriction modification system and the first evidence for the existence of molecular scissors that actually cut DNA. And he showed that E. coli bacteria have an enzymatic immune system that recognizes and destroys foreign DNA, but modifies the native DNA to prevent self-destruction. This is a very primitive immune system that we are talking about. Once this discovery was made, and Werner Arber and Daisy dusswa actually published those papers, a lot of people at that time in the 50s and 60s who were working with bacteria, different kind of bacteria, went back to the labs and grew these bacteria in large fermenters and actually isolated these enzymes that Werner Arber and Daisy dusswa mentioned in the first papers. And two among the many people were Dan Nathans and Hamilton Smith at the Johns Hopkins University in the United States, who did some very, very pioneering work on purified restriction enzymes that Werner Arber actually suggested that they occur in E. coli. And in 1978, all three of them, Werner Arber, Daniel Nathans, and Hamilton Smith, were awarded the Nobel Prize in Physiology or Medicine for the discovery of restriction enzymes and the potential or the application to do a lot of nice molecular genetics. I think that the discovery of these enzymes that you can cut DNA and later on, you have an enzyme that actually puts DNA together by an enzyme called DNA ligase. The birth of biotechnology or genetic engineering came. And if you have to summarize biotechnology in one or two words, it is cut and paste, cut DNA and paste DNA. And that is all there is to biotechnology. Whether you do it in bacteria, whether you do it in fungi, whether you do it in plants, or you take mammalian cells or human cells, DNA is very, very similar in its chemical composition. And therefore, you need enzymes that cut DNA. And once you cut DNA, you just have to take that piece of DNA that you're interested and put it into a vector, which I'll talk about it a bit later on. And then you can amplify or multiply that particular piece of DNA that you're interested. So it's cutting and pasting that you are talking about in biotechnology. And that is what is genetic engineering all about. Whether, as I said, whether you do it in plants or animals, it doesn't matter at all. This is Werner Arber, for a couple of years ago at the Biocentrum in Switzerland, because the discovery was made in Geneva, but now he's in Basel. And the next slide shows you the two scientists from Johns Hopkins, Dan Nathans and Hamilton Smith. Hamilton Smith is a taller person who actually did some nice in vitro experiments, taking purified DNA, taking the pure enzymes, cutting it, and showed that these enzymes are very, very specific. They recognize a particular sequence on the DNA, and once they recognize the sequence of the DNA, they will cut. So every bacterium has at least one restriction enzyme. And this is how the bacteria protect themselves against the phages of bacterial virus. If a foreign DNA comes in, the foreign DNA is actually chopped off by the host enzyme, and the host DNA is itself protected because there's another equivalent enzyme called the modification enzyme, the M, which modifies its own DNA, and therefore the restriction enzyme cannot act at all. Now, this is a particular slide that I want to talk about a little bit. This is Daisy dusswa, who was a lab assistant in Werner Arber's lab in Geneva, when the famous discovery of restriction modification of enzymes were done. Now, there's some controversy about this, that many people felt that Daisy dusswa should have got the Nobel Prize along with Werner Arbor, but that didn't happen. She was a lab assistant, and of course, Werner Arbor had all the ideas, and these were actually carried out by both of them. But unfortunately for Daisy dusswa, when she moved out of Geneva and went to San Francisco, she worked with another person called Harold Wormers, who went on to get the Nobel Prize in 1989. Again, Daisy dusswa missed out on the Nobel Prize because of this one person who got the award. And then in 1980, she came back to Paris. She headed one of the laboratories at the Pasteur Institute, and in 2014 she actually passed away. This is one of the quirks in life, that a lot of deserving people have not got the Nobel Prize or the recognition. And in particular, I bring this point of Daisy dusswa because it's something like Rosalind Franklin, along with Watson and Crick, should have got the Nobel Prize or should have been named for the Nobel Prize. But she was not named because she passed away. She died before the announcement. I think the Nobel Prize Society committee, there's a rule that when you're not alive, you cannot be given the Nobel Prize. So posthumously, the Nobel Prize is never given. But I think Rosalind Franklin really deserved it and missed out. And here's another example of Daisy dusswa, where many people felt, because of the critical experiments that she did with Werner Arbor, actually deserved some recognition. So this is what RM phenomena is. You can see from this slide here that the phage actually is entering here. The phage injects its DNA into the cell, and the cell has this host enzymes called restriction enzyme and modification enzyme. The modification enzyme of the host actually modifies the DNA, puts a methyl group either on adenine or cytosine, and the host DNA is completely modified. Whereas the restriction enzyme, which acts on any kind of DNA, either host DNA or the phage DNA, foreign DNA, will act only on unmodified DNA because the phage DNA is not modified at all. It's unmodified, and therefore the molecular scissor will chop it off and will not allow the phage to multiply. Whereas the same restriction enzyme cannot act on its host DNA because the target DNA is completely modified or methylated, and therefore this phenomena is known as restriction modification. And now you can appreciate that this is a primitive immune system. It is a thing called self and non-self. The self DNA is protected by methylation, whereas the foreign DNA is the non-self, and therefore it gets chopped off. So the major purpose of these RM systems, which was the discovery of Werner Arbor and Daisy dusswa, is a defense system. In the bacterial world, in the evolution, when life started, this is a very primitive immune system. You and I have T cells, B cells, and all sorts of cells to take care of our defense against pathogens. But the bacterial world, this resembles the immune system, the R and M. The R chops off the foreign DNA, whereas the M protects the host DNA, therefore the R cannot act on it. So if you have all understood this RM phenomena, I think that is half the exercise that I want to tell you, that this is a major discovery in biology. And once Werner Arbor and Daisy dusswa came out with this, as I said, Dan Nathans and Hamilton Smith went on, and many other people went on to purify enzymes, and now you have them all commercially available. You can buy these enzymes. Each bacterium has at least one restriction enzyme. I'll tell you later on that some bacteria have more than one restriction modification. So the major purpose of the RM system, as you see here, is in evolution rather than in the vital cellular processes. Because once the foreign DNA is actually chopped off into pieces, these small pieces of DNA can be integrated into this host chromosome by methods of genetic recombination, and then you can get different kinds of species and things like that. So the role of RM systems in vivo, inside the cell, is actually in the process of, helps in evolution processes and things like that. So restriction enzymes, just to tell you very quickly, are enzymes that naturally occur in bacteria, and the bacteria use these enzymes to protect themselves from viruses, that is bacteriophages, that are normally invading them. And restriction enzymes cut up the DNA that the virus injects into the bacteria so that the virus cannot be reproduced. Now, one question that some of you will ask me, what are those one or two colony plaques that the E. coli B strain had? Because some phages survived. Because they survived, they form a plaque. How do those happen? And I'll come towards the end of my talk, that even viruses or the bacteriophages over a period of evolution have devised anti-restriction strategies to counteract the host restriction enzymes. And therefore, you see this battle between the host bacteria and the viruses. At one point, the viruses are above the bacteria, and at other times in evolution, the bacteria have evolved, and therefore they do not allow the viruses to multiply and things like that. So this arms race between the bacterial virus or bacteriophages and bacteria is all the time happening. And therefore, for especially students, I want the teachers to tell the students that studying simple bacterial systems can give you a wealth of information. And, it's the basis of all phenomena that you see in eukaryotic systems. But in bacterial systems, it is very simple, but the same chemistry, whether in replication or transcription or recombination, everything is actually from the E. coli studies that people have done. So what is true for the E. coli is also true for the elephant. This is a famous adage that goes on. Is this clear? Hello?
Yes, sir. Yes, sir. It's clear, sir.
Okay, I'll continue. All right. So if you take an enzyme called EcoR1, which is the most famous, well-known restriction enzyme, it's a restriction enzyme that is derived from E. coli, and therefore the E and the C and the O come from there. R1 is the serotype and is the first enzyme that was discovered in this bacteria, and therefore it's called EcoR1. So as I said, the restriction enzymes like EcoR1 recognize a very specific sequence. They recognize a very specific sequence. And this is the sequence. The sequence is GAATTC. But it has to be in the double stranded. And in the opposite strand, it's in the reverse direction, GAATTC. So this set of sequences is actually a twofold axis of symmetry. And this is called a typical palindromic sequence, where you read it from left to right, GAATTC, and then from right to left is GAATTC. This is just like the word Malayalam. If you read Malayalam from the forward direction or from the backward direction, it will be the same. So these are palindromic sequences, and many restriction enzymes recognize palindromic sequences and cut the DNA. That means they cut the phosphodiester bond between GNA and GNA. And therefore, when you treat it with the enzyme, this DNA is now broken into two pieces. And if you follow these blue arrows here, you can see these two fragments are created and the enzyme cuts DNA. Now, if you cut some other piece of DNA with the same enzyme, you will get the same kind of ends that you have here. And now you can mix this one and the other one, and then you can join it. A phosphodiester bond is made by an enzyme called DNA ligase, and then you have a recombinant DNA. This is everything that you want to know about biotechnology or recombinant DNA technology, and it's possible only because of the discovery of Werner Arbor and others, where they found these enzymes which can cut DNA. And again, an enzyme like DNA ligase, which joins the fragments, form the basis of the biotechnology that you're talking about. Now, here are examples of many restriction enzymes. I've just shown EcoR1, we talked about GAATTC, and it cuts here and here, so therefore you have something called a end which is not blunt. Whereas if you look at an enzyme called HAE3, this is from Haemophilus aegyptus, a bacterium. It cuts right in the center so that you have two GGCC and CCGG. So you have something called sticky ends and blunt ends, and you have a number of enzymes. And I tell you right now, as of today, we have about 5,000 restriction enzymes that people have actually characterized or identified. Many of them have been purified, and lots of companies sell these enzymes so that you can use these enzymes to cut DNA. If you have a DNA and it has this particular sequence, you can use EcoR1. If the sequence has HA3, you'll have a sequence called GGCC. So any piece of DNA can be cut by a battery of these enzymes. And this is what I've shown here, that once you cut it, you can run it on an agarose gel electrophoresis, and fragments of DNA with different sizes will run. You cut it with restriction enzyme A, restriction enzyme B, restriction enzyme C. Then you measure all these sizes, and you can actually create a restriction map, which is shown here. So any genome, and this is how the human genome was actually sequenced. First, they had to chop it off into a number of fragments by using restriction enzymes, and then sequencing these fragments and assembling it together to form the whole human genome. This is a prokaryotic genome, an E. coli genome, and you can see by using a battery of enzymes, enzymes like RSR2, SMAR1, all sorts of things, you can create a whole restriction map here, and you have an idea of how the genome of a living organism looks like. You can do it with plant genomes, you can do it with mammalian genomes, Drosophila genomes. Every genome that has been sequenced, and every day almost 10 to 15 sequences are coming out, the use of restriction enzymes becomes so important, and therefore the discovery by Werner Arber and Daisy Dusswa is very, very significant. This is a company in the US called the New England Biolabs, and they make the best enzymes. They make a lot of enzymes. In India, we have a company called Bangalore Genie, which makes a few enzymes, but the New England Biolabs is one big private company which makes huge amounts of restriction enzymes and good restriction enzymes. It was actually founded by this person called Donald Combe, who's on the right side. And this scientist on the left is Rich Roberts, who was in the Cold Spring Harbor, who was actually the first one to isolate some of these enzymes. Johns Hopkins scientists also isolated, but Rich Roberts made a lot more enzymes. And he and Don Combs both got together and started this company called New England Biolabs. A fantastic lab. I've been there a number of times, and both these scientists have predominantly dominated the whole area of restriction modification enzymes. Rich Roberts, who was in Cold Spring Harbor and went and joined the New England Biolabs, actually years later got the Nobel Prize for something else altogether. This is on splicing of eukaryotic genome, but that is where the greatness of that man is. Now, once Werner Harper published his papers and Dan Nathans and Hamilton Smith isolated a few enzymes, a lot of restriction enzymes. As I said, about 5,000 enzymes have been actually identified, many of them purified, and a lot of biochemistry has been done. And like all books that we have, these enzymes are actually classified into type 1, type 2, type 3, type 4. I don't think this classification is important, but in all our school books, college books, we have this classification. And the classification is based on a number of properties of these enzymes. You must remember that all these enzymes cut DNA, and they all cut DNA specifically. They recognize a specific sequence and cut DNA. But this classification has come into picture because of all these properties on the left, because of the subunit structures, the enzyme activity, the biochemical cofactors, the recognition sequence, the cleavage site, and many other properties. These have been divided. But for our lecture now, we will concentrate on the type 2 enzymes, the second column here. These are the simplest restriction enzymes in their composition. They recognize a specific sequence and cut within the sequence. GAA, TTC, they cut between G and A. Whereas the type 3 enzymes on the next one, these are also restriction enzymes. They also recognize specific sequence, but they don't recognize palindromic sequence. These, recognize palindromic sequence, whereas the type 3s don't. And they cut away from the sequence. They recognize one sequence and cut away. So it's like an action at distance. And therefore, these have not become very popular for cloning and biotechnology purpose. But the set of enzymes which are called as the type 1 enzymes are the most complex enzymes. They also recognize specific sequence and cut sequence, but they cut 1,000 to 7,000 base pairs away from the recognition sequence. But every bacterium, you should remember, has in its chromosome at least one type 1 restriction enzyme. So that is the major enzyme for defense purposes. All the type 2 enzymes that talk, like EcoR1, Hindi3, BAMH1, they're all extra chromosomal coded, and therefore they're not part of the host chromosome. But the chromosome, the bacterial cell hosts something called episomes and things like that, which actually carry the type 2 enzymes. So you have type 3, type 4. I'm not going to go into detail. Just to complicate a little bit, within the type 2, you have type 2S, type 2E, type 2F, type 2T, type 2G. And so this classification is actually not very useful. I don't think, because, you have enzymes like the typical type 2 enzymes which cut within the sequence. Like EcoR1, if you see here, it cuts within the recognition sequence. You take type 2S, which I'll talk about them later on. A good example is FOC1. It also recognizes a specific sequence, but cuts away from the sequence. 9 nucleotides away from the main sequence. Type 3 cut 25 base pairs away. Type 1 cuts 1,000 to 7,000 base pairs. So among the type 2 only, you have lots of variations. And therefore, I think classification of enzymes is actually meaningless. There's a whole continuum of enzymes that take place here. Now, all this work, the biochemistry and the use of these enzymes in biotechnology, which I'll discuss later on, were all done, are being done. But in 1995, when the first free living organism was sequenced, the whole genome was sequenced, and that organism is Haemophilus influenza. And when they sequenced it, they found that there were a number of open reading frames in the Haemophilus genome. Each of these open reading frames, which I've shown here, look like restriction modification enzymes. This is by sequence analysis. The computational biologists, bioinformatics can compare sequences and then say there's a similar sequence in this organism or that organism. So by computation analysis, it became very clear that Haemophilus influenza has a number of open reading frames which look like RM systems. But biochemically, before this discovery, at least 3 different kinds of enzymes were isolated from Haemophilus influenza. They were called as the HIND3, HIND2, HIND1, HIND1, HIND2, HIND3. But now you have HIND4, HIND5, HIND6, HIND7, HIND8. So a lot of new information came out from these sequencing projects. And remember, this is the first sequence of a free living organism. And the sequencing was done because the Haemophilus, how was it done? The Haemophilus influenza genome was cut with restriction enzymes to the number of fragments. Each of the fragments was sequenced and then pasted together, joined together, and then you have a genome. So again, here you have the usefulness of restriction enzymes. Here is a slide which shows you Mycobacterium tuberculosis, the genome. This is the organism that causes tuberculosis, which is very prevalent in India. This bug also carries a lot of open reading frames, which look like restriction modification enzymes. And it is now, ever since these papers came out, were published, a lot of people have actually isolated many of these enzymes. On the top, I've given the genome. I mean, it's an organism called Methanococcus yanashi. This is an organism that grows at 95 degrees centigrade in the hot springs. And remember that when this genome was sequenced, it was found that the Methanococcus yanashi has a lot of open reading frames, which look like restriction modification systems. What is the purpose of RM systems? It's a defense mechanism so that when the phage comes in, the phage DNA is actually chopped off. But if this organism grows at 95 degrees centigrade, the chances of any phage infection is very, very low. If that is true, why does this genome have so many restriction modification enzymes? And therefore, the first idea, other than the defense mechanism, the hypothesis was this that these enzymes may be present for some other purpose that we still do not understand. Is this clear? So Methanococcus yanashi is an interesting organism. Here is an organism of a very famous strain called Helicobacter pylori. This is a bacterium that causes ulcers, gastritis, and is the only bacterium that causes cancer. And all of us in India, 60% of the world population in their stomach have this bacterium. It's a harmless bacterium. But if it becomes pathogenic because of mutations, it can cause ulcers, it can cause cancer, it can cause gastritis. And therefore, when the Helicobacter pylori genome was sequenced in 1999 2000, lo and behold, you find that there are 44 open reading frames which look like restriction modification. I couldn't accommodate both of them in one slide, but each of them looks like an RM system. And I keep saying looks because many times you'll actually have to do experiments to show that this actually is an enzyme with a nucleus activity or with a modification activity. And my laboratory has been working on these enzymes, and therefore in 2007, we started looking at H. pylori restriction modification enzymes. Very nice piece of work that a lot of my students have done. But just to tell you that these enzymes, which are present in the Helicobacter pylori, have a purpose other than defense mechanism. Again, this bacteria survives in acidic conditions in the stomach, pH 1 and pH 2. The chances of a phage infection is very, very unlikely, although there's one reported case of a phage specific for Helicobacter pylori. And therefore, my laboratory and several other laboratories in the world are looking at what are the functions of these RM systems. It is not just for killing phages, they must have some other purpose altogether. Just to quickly tell you what in my lab, and I'll summarize in this one slide of my lab's work, that we find that in H. pylori, new RM systems are made, they're maintained for some time, they are inactivated, and they're thrown out. And then you have again new enzymes forming, they're maintaining. So all the time the H. pylori is making RM systems. And this is just to survive in the acidic conditions of the stomach. That there are huge amounts of variations, mutations that accumulate in pylori, and these mutations is what results in a dynamic and evolving system of RM systems. So very nice information that has come out by studying RM systems, although the whole purpose is in biotechnology, and as most of you would like to use them in biotechnology, but the biology itself of this organism is very interesting. And the H. pylori genome has 44 or 45 open reading frames which look like RM system. Therefore, we must give some importance as to what these systems are doing in a bacterium called H. pylori. I will move on now, and just to clarify, just to be sure, Is everybody following? Or if there are any questions that you can quickly ask, then I can proceed.
So there are two questions asked by participants. We will take questions and answer. Yes, sir. Shall we take now or
Okay.
we can take at the end of the session, sir?
All right. Okay. All right.
Okay, sir.
Very good. Now I talked to you about type 2 restriction enzymes. That is enzymes that recognize a sequence and cut within the sequence. But you see, Werner Harper, when he did the experiments, he did with much more complex enzymes called the type 1 enzymes. And here I've given you, these are all various type 1 enzymes. You can see the recognition sequence is not simple. It's not a two-fold axis of symmetry. They're all asymmetric sequences. And more than anything else, the type 1 enzymes, again, can be classified into A, B, C, and D. We won't worry about that. But these enzymes recognize this sequence and cut 1,000 to 7,000 base pairs away from the sequence. Good examples of action at a distance. I bring this particular slide because to tell you that every bacterium host has at least one type 1 restriction enzyme, which is encoded by the chromosome, the host chromosome. And if we look at the structure of these enzymes, these have been purified also by a variety of people. And it's a huge protein. It has so many subunits, and all these subunits have to come together, and then it recognizes a specific sequence and then cuts it. So this is the most complex of the restriction enzymes, although they were the first ones to be discovered, the type 1 enzymes. And the very interesting properties of it, which we don't have time, but to talk about them, but they're absolutely fascinating set of enzymes that you have here. I'll come back to the bottom part of this slide later on, but I want to leave you with this thing that the type 1 enzymes are the most complex, the type 2 are the most simplest, and the type 3 enzymes, which my laboratory has been working for the last 30 years, are somewhere in between the type 1s and the type 3s. Now, like the type 1s, the type 3 enzymes also are slightly complex. And they're complex in the sense that for an enzyme to cut via type 3 enzymes, you require two sequences which have to be in the opposite orientation. If they're in the same orientation, the enzyme will not cut. The recognition sequences should be in the opposite orientation. And therefore, the restriction enzyme, which is shown here in red and blue, binds to the sequence. Another molecule of the enzyme binds to the other sequence, and the DNA in between is actually looped out. As you can see here, there is DNA translocation. And for this, you require energy, and therefore ATP is a must. ATP hydrolysis is a feature of type 3 and type 1 enzymes. The type 2 enzyme does require magnesium for cutting, whereas here, the way these enzymes cut, there's DNA translocation, the loops are brought together, and then there is cleavage, and you have two fragments, you have two pieces of fragment. This is a model that was proposed from very simple biochemical experiments. And therefore, everybody believed that these loops should be formed, but for about 25 years, there was no evidence of this loop at all. So my laboratory, in conjunction with one of the most powerful microscopes, the atomic force microscope made by Olympus, and the University of Cambridge collaborated and showed that the type 3 enzymes do form loops. You can see here, the loops are formed. We measured the distances, and as the two complexes come together, the loops become bigger, and we measured the loops and showed that this was an experimental evidence to show that DNA looping by the type 3 enzymes is an inherent feature of these enzymes. Unlike the type 2 enzymes, which bind and thus cut DNA, these guys cut DNA after getting two sequences together or thereby. So this is, again, I want to highlight a lot of physics here and optical microscopy imaging. One should have some little knowledge about little physics, a lot of chemistry, and then biology becomes very, very interesting. You should imagine by now that the combination of this makes this whole area an interdisciplinary area. The biochemistry is highly interdisciplinary and things like that. Let's get that. I'll skip that. So we now come to the biotechnology. So all forms of genetic engineering revolve around the production of recombinant DNA and the use of restriction enzyme, because the restriction enzymes cut the DNA. An enzyme called DNA ligase will combine DNA, and therefore you have a recombinant DNA. And this whole technology of cutting and pasting is now what is biotechnology. And I think students, nowadays students are very good in cutting and pasting, especially when you have to write assignments and all. They cut and paste from the websites. But I think, this is what biotechnology or genetic engineering is all about. Recombinant DNA technology, whether you do it in bacteria, whether you do it in fungi or plants or mammals, is the same. It's cutting DNA by restriction enzyme and pasting DNA by DNA ligase. So when you say cut DNA, it's actually a phosphodiester bond which is cleaved. And when you say the DNA is pasted, you're creating a phosphodiester bond. So cutting, hydrolysis of a phosphodiester bond and synthesis of a phosphodiester bond is what is biotechnology all about. And this is what I want to show you here. Here, this is a blue sequence and this is a pink sequence. Both have a recognition site for EcoR1. So you cut this DNA with EcoR1, you will get these fragments. You cut this DNA with EcoR1, you get these two pink fragments. And now mix all four of them and then add the enzyme called DNA, and you get a hybrid, which is a recombinant DNA molecule. This is it. This is recombinant DNA technology. Whether you do plant biotechnology, you do animal biotechnology, the business end of the whole process is cutting and pasting DNA. And once you know the chemistry and how these enzymes work, you can make lots and lots of recombinant DNA molecules. And that is what recombinant DNA is. A new DNA sequence is created when the DNA of one organism is inserted into the DNA of another organism. And this combination, new combination of DNA, is known as recombinant DNA. And this is all because of the discovery of restriction enzymes and the other enzyme called DNA ligase. And this is one of these slides I want to show you. So you take a cloning vector where you want to multiply a piece of DNA. Both the cloning vector and the foreign DNA are cut by the same enzyme, say for example EcoR1, and you get something like this. And you get three pieces here, and you are interested in the middle piece, not because of your DNA is here, your sequence is here. Now you take all this, isolate it on agarose gel electrophore because of size, and then mix this and this, and you get a hybrid molecule. So this is the basis of recombinant DNA, right? This is how you make recombinant molecules. And this particular one, again, is shown here by another site. There's a plasmid, which has one EcoR1 site. This is a foreign DNA, and this is the DNA that you're interested in cloning. The genetic information, say for example, a protein of interest is encoded within the EcoR1 sequences. You cut the EcoR1 by EcoR1, cut this by EcoR1, join them together by ligase, and you have a put it back in the bacteria, and you have a recombinant DNA molecules. And this is what these three great scientists actually did. These are the actual pioneers in recombinant DNA technology. Paul Berg, Stanley Kohut, and Herbert Boyer, who actually went on to form a company called Genentech, which is the most famous and most famous company of biotechnology companies in the world. This is in San Francisco. They all Herbert Boyer actually found a businessman called Bob Swanson, who provided the money. And this is what they did. Simple, they take a plasmid, cut it, put a synthetic DNA, and made a hybrid molecule. And the whole company, Genentech, was formed. Genentech actually was found, the laboratory was in the basement of a house. And now in San Francisco, in the Bay Area, the most poshest place in the world, you have 42 buildings, which is Genentech. So they make a lot of recombinant DNA molecules. And remember, this is where the restricted enzymes come into so much of use. The company was founded in 76. by venture capitalist Bob Swanson and the biochemist Herbert Boyer. And they first successfully expressed the human gene in bacteria when the hormone called somatostatin was actually cut with certain enzymes and then inserted into the bacterial plasmid, and you can make lots and lots of somatostatin. Like insulin, for example, insulin which many diabetic patients use. The source of insulin is the pancreas. The pancreas secrete a lot of insulin. They make insulin and secrete a lot of insulin. And traditionally, this insulin was got from pigs. A lot of pigs had to be killed. The pancreas had to be taken out. And from the pancreas, they produced insulin. They purified insulin. But now if you go back and look at recombinant DNA technology, you can actually take one pig, for example, take out the pancreas, isolate the insulin gene, which is very easy to do nowadays, and take that insulin gene and put it into bacteria. And therefore, the bacteria multiply every 20 minutes, and therefore you can have tons and tons of insulin. And that is what now most of the insulin made that is available is recombinant insulin. And you see, even in India, Biocon produces insulin. A number of companies produce this, but the first set of experiments were done by Herbert Boyer in Genentech, and therefore they are actually the forerunners of biotechnology in general. So what is biotechnology? Is a manipulation of living organisms and organic material to serve human needs. You can use it to make insulin. You can make it to do many things. You can clone plants. You can clone animals. Artificial insemination is also a biotechnology process. Yeast, which make bread, for example, or in alcohol production, you can modify the yeast you can make recombinant yeast, and therefore a lot of applications are now possible, and people are exploiting this particular technology. So why it becomes important for us to now, realize what is the relationship between science and technology. The experiments that Werner Arber, Daisy Dusswa, or even the in vitro experiments done by Hamilton Smith and Dan Nathans is purely science. So science is the search for knowledge about ourselves and the physical world. The process is a scientific method looking for patterns in physical world, evaluated by how well facts support conclusions, and limited by ability to collect evidence. And you make discoveries. And it is these discoveries that give rise to technological advances. And therefore, it comes down to technology. Here is a case of restriction enzymes, which was shown in bacteria. And now you have a practical application of knowledge. And you adapt ourselves to physical world. The process is finding solution to problems like insulin. You cannot be killing a lot of pigs and pancreas, but by cloning and by doing recombinant DNA technology, there's a solution to this problem of mass production of insulin and things like that. So science and technology go hand in hand. And you cannot always emphasize that technology is more useful than, because because of the work done in science, the technology actually progresses. It's not only true in the biology, in every subject that you talk about, basic sciences become very, very important. And as teachers of graduate and undergraduate students, I think you should emphasize this, that the students should know basic knowledge, should have the concept very clear. I mean, biotechnology is a huge area, but if you actually cut it down, it is cutting and pasting. That's all. You cut the DNA and paste DNA, and you have biotechnology. So I've written down a few things because I just wanted to remember, but I think just to tell you that you're cutting DNA and pasting and all, but in nature, this has been happening. Nature has been employing different variety of crops and all that you see, rice yielding and all that. Naturally, you have recombination going on. And therefore, biotechnology is actually a process that nature has actually employed. and doing it. But we now using physical methods and things like that, we're trying to modify living organisms according to human purposes, to our purposes, and things like that. So the American Chemical Society defines biotechnology as the application of biological organisms or systems or processes by various industries to learning about the science of life and the improvement of the value of materials and organisms such as pharmaceuticals, crops, and livestock. So one can talk a lot about biotechnology, but I think you must remember that simple microbiology experiments actually led to this, the whole technology. You can produce genetically modified plants and animals by inserting desirable genes from one organism into the DNA of another organisms. Make plants insect resistant. I think farmers will like this very much. Make plants which are salt resistant, especially in the mushrooms, the mango groves, and where you have a lot of salt and things like that. Plants are normally susceptible for destruction, but if you make them salt resistant, they will survive. Or when in a tsunami region, because you have a lot of salt and things like that. You can make many other things. For example, you can make mice or bacteria glow in the dark by putting a fluorescent protein, which I'll show you in this. So this is again genetically modified organisms which are consumed by millions of people. For example, this is soybean here. You can modify it and you make it rich in amino acids, rich in something by doing simple recombinant DNA technology. So this is genetic engineering. Recombinant DNA technology is one and the same. And I think it is a question of deleting or adding or replacing genes. You can make chicken without a feather, which will be very, very useful when you want to eat chicken, for example. You don't spend time taking off the feathers. Here, this is a featherless chicken that is possible. You can create animals, organisms that glow in the dark that you can't see in the natural light. But because you have now cloned a fluorescent protein, if you now flash UV, for example, you can get this. Here are mice which are expressing a protein called the green fluorescence protein. And they all become green when you flash UV light on things like that. So we have now technology, we can introduce genes, we can delete genes. In other words, we can play with nature. And therefore, there's a lot of regulatory procedures now. You can't do many of these experiments in human beings, it's not allowed at all. Any kind of recombinant DNA technology experiment that you're planning to do requires the approval of a number of regulatory agencies. The Department of Biotechnology, DBT, has this whole process in line, and therefore one can do these experiments to a certain extent. You can produce medicines like insulin and the human growth hormone. And this is what the initial experiments of Genentech were. And you get insulin in the form of Humulin, Novolin, various companies making recombinant DNA insulin. No more killing pigs, no more getting the pancreas out of them and things like that. You can solve crimes, for example. I think this is where in DNA fingerprinting and forensic science that biotechnology has come in a big way. By isolating DNA from any kind of a fluid, you can actually sequence it and find out and figure out who is the person, especially when the no two people have the exact same DNA sequence. So everyone's DNA fingerprint is different. And especially at the crime scene, for example, or when a rape scene or a murder scene, for example, isolating DNA from hair or from any of the body fluids, you can actually identify who the person is. And this is by, again, isolating DNA, cutting it down with restriction enzyme to various fragments and sequence it. And you can actually create a link between the person to a crime and things like that. In many hospitals, especially in India, for example, the babies get mixed up when they're born. And therefore, checking the mother's DNA and the father's DNA, you can now sort out this whole process, because by just restricted enzymes, you can create a pattern, and the pattern will be a combination of mother and father in the case of a child and things like that. So when you use this, and I think all of you read a lot about this in newspapers and things like that, but I think biotechnology has become a branch by itself, a subject by itself. Bioinformatics, the computational biology portion becomes very important here. Genetic engineering is a starting, what we started off with engineering genetic material, for example, used in diagnostic, fermentation technology, food innovation, energy, environment, animal agriculture. I mean, you talk about anything now, you can use biotechnology. And biotechnology is, the use of these enzymes, which become very important. So to summarize, science drives technology and technology drives science. Biotechnology has been around as long as humans have practiced agriculture, so there's nothing new. Biotechnology is used to conduct research, develop new products, and improve processes. And DNA-based technologies have been developed in microbes, and as a society, we should be responsible for using it wisely. Because you can use always this in the wrong sense, and therefore there's a lot of regulatory bodies which control what kind of experiments you're allowed to do. You must get permission from them and do it. Do I have more time or shall I end it here, Ramesh?
10 more minutes, sir.
Huh?
You have 10 more minutes, sir.
10 more minutes. Okay, I will
Yes.
talk for 10 more minutes.
Yes, sir.
So this, as far as the restriction enzymes and biotechnology are concerned, I have covered what I wanted to say. But I thought I will tell the students and the teachers something new. And remember the RM systems that were discovered by Werner Arber was in the 50s, late 50s, 60 years, early 60s. Exactly 50 years later, in 2000 or 2010 now, again people focused on bacteria because of this very unusual system that they discovered. Very briefly to tell you, when a phage or a bacterial virus invades a bacterium, it has to be absorbed. It should absorb itself on the surface of the bacterial membrane. Then the DNA has to enter here. Then the DNA of the phage should multiply. It has to be transcribed, make proteins on its own. It has to assemble small phages and then kill the cell, and the phages are completely out of it and they can go back and do this. This is a typical cycle of a phage. Now, restriction enzymes that I talked about come in this stage, that as soon as DNA is entered, the DNA is gets entry to the thing, the host restriction enzyme will chop it off, and therefore the phages cannot multiply anymore. But if in case some phages do multiply, then they will go through replication, transcription, and all this process, things like that. So you can block phage. You can block at the adsorption stage, blocking the phage receptor there. You can produce a competitive inhibitor for these kind of things. Abortive infection. You can prevent phage DNA entry by restriction enzyme. And that is where Werner Arber made this discovery. And now, in the last 10, 15 years, this whole thing called CRISPR-Cas, which has come up in a big way, again comes from the discovery from phages and bacteria. So studying phage bacterial interaction is again come back into a big light, and now any year, I think very quickly, the people who worked on this CRISPR-Cas or something equivalent are all tipped to become the Nobel Prize winners, maybe this October or very soon. So I thought I'll spend a little time on these systems. Again, to tell you or highlight the point that how restriction enzymes became so useful and everything came from bacteria, the CRISPR-Cas systems also come from bacteria and phage. And therefore, studying prokaryotic systems is becoming, is always a fascinating area to study this. So this is the area called genome editing or gene editing. So it's a process whereby researchers can introduce a modification into an endogenous gene. You modify that, or you disrupt it, you insert it, you replace it at a locus in the genome, which then controls the gene expression, creates a single nucleotide polymorphism, and create reporter fusions while maintaining the endogenous gene regulation. So suppose you want to knock out a particular gene. For example, in hemophilia, for example, that the hemoglobin genes, for example, there's a single amino acid mutation which makes it from normal to a sickle cell. I'm sorry, I'm talking about hemoglobin, not hemophilia. Hemoglobin. So there, sickle cell hemophilia. I'm sorry, hemophilia. Yeah. So one amino acid change will make the hemoglobin less effective in terms of oxygen transport and things like that. And now you want to correct it. Earlier we still have this whole process called gene therapy, but it was completely banned because of one unfortunate case. How do you replace this defective hemoglobin gene? By changing one amino acid there, one nucleotide change, and therefore the amino acid will change, and you rectify it in the before the baby is born and things like that. So this is where genome editing becomes very useful, that you're editing the genome by modifying it or by doing something so that the genome is altered. You can do it in a plant, and therefore you can make a variety of plants. You can edit the plant genomes, animal genomes, bacterial genomes, what have you. 3 common targeted genome editing systems are now known. One is called the zinc finger nucleases. These are again enzymes. One is called talons, which is also a protein-based, protein-guided nuclease. And the third one, which has attracted a lot of attention, is this RNA-guided CRISPR-Cas system. All 3 of them actually come from work done on bacteria and phages. And I thought I will mention about these 3 very quickly in the next 5 minutes. That the zinc finger nucleases were the first ones. These enzymes, they're not naturally found, but these were created, which can be used to edit the genome. You can make corrections in the genome, and therefore it becomes very important. And this work on zinc finger nucleases actually comes from the restriction enzymes, the type 2 restriction enzymes. I told you about EcoR1 recognizes a sequence and cut within the sequence, right? So that's what it is. Whereas you have an enzyme in the type 2S, which recognizes a sequence and cuts 9 nucleotides or 14 nucleotides away from the sequence. This is action that's a distance. So you have recognition in one part of it, and cutting is different from it. And therefore, this enzyme has 2 domains called the fork1. One is the DNA binding domain, and one is the domain responsible for cutting. And these domains are at the N terminus of a protein and the C terminus of a protein. And an Indian scientist by the name Srinivasan Chandrasekaran in Johns Hopkins, working in Hamilton Smith, who got the Nobel Prize, actually showed that the fork1 has these 2 domains. And he asked the question, if I take the DNA binding domain of fork1 or any DNA binding, lots of proteins bind to DNA, and therefore, if I can isolate the DNA binding domain of a protein and fuse it with the nuclease of fork1, then I can make an enzyme which was not there before, and wherever the DNA binding site is, I can put various DNA binding domains, fuse it with the nucleus, and then I can edit genomes. And this is the first set of gene editing nucleases called the zinc finger nucleases. There are some proteins which bind to zinc, they have a zinc binding domain, which are important for DNA binding. And he actually fused a number of zinc finger proteins to the nucleus and made a nuclease which now cuts DNA at where you want, where you desire it to cut. This is different from restricted enzyme, right? Restricted enzyme cut at the recognition sequence and then. Whereas you're now putting the DNA binding domain and asking it to cut at the place that you want. And therefore, the zinc finger nucleases is a very important discovery in the whole history of these gene editing nucleases. And Srinivasan Chandrasekharan, who's actually from Pondicherry, went about 40, 45 years back went to the US 40, 45 years ago at Johns Hopkins and did this work. I bring this particular point because the next set of nucleases that became quite popular were called the TALENs, called the transcription activator like effector nucleases. This is again from another bacteria called Xanthomonas bacteria, where the DNA binding was actually isolated. What Chandrasekharan did was zinc finger domains, but this is a different kind of a DNA domain, which was again fused to the FOC1 nucleus. And they created gene editing nucleases. We won't go into details of this. So these are called talents, the tail and the nucleases. And this is what this paper came out in 2010 in Science, whereas the zinc finger nucleases were done in 1994 by Chandra and others. And now we come to this, one of the most hot areas in biology is the discovery of this CRISPR-Cas systems, which again comes from bacteria and phage origins. Again, these are gene editing nucleases. The Cas is actually a nucleus, whereas the CRISPR is a sequence of DNA, a combination of DNA sequence from the phage and from the bacteria. And when phages enter the host DNA, the host DNA already has some sequences which resemble that phage DNA, and the Cas nucleus will now chop it off so far that the phage DNA does not multiply. I won't describe, but this is the whole procedure. But just to tell you the point, that again, this discovery in 2010 comes from bacteria and phage, and therefore studying these microorganisms becomes very important. So if you compare the zinc finger nucleases, which came in 1996, the talons, which came in 2010, and the CRISPR-Cas now come 2012, but this is the most widely used gene editing nucleases now. And just to tell you, this is, in 2012 it was discovered, and see the number of publications at 2015, they're just shooting up. Lots, every paper that you now find in literature has some kind of a nucleus, gene editing nucleus by CRISPR-Cas that people have used. A lot of controversy in this as to who first discovered this CRISPR-Cas. There are two sets of things and a lot of patent applications and a fight between MIT and the University of California, the two scientists. And both the scientists are tipped for the Nobel Prize for the discovery of CRISPR-Cas. But my point here is to tell you that CRISPR-Cas is a genome editing nucleus, but the concept came from Chandra's work on the discovery of zinc finger nucleases. So one has to keep in mind, and hopefully the Nobel Committee keeps in mind when they talk about history and to actually find out where the concept is. Although CRISPR-Cas is much easier to use and a lot more people have used, but the concept actually comes from Chandra's work on zinc finger nucleases. So there's a huge craze about CRISPR and Cas, because you can now do editing, you can do genome editing, you can rectify disease conditions before in the womb, before the baby is born and things like that. So it's actually heavily used in gene therapy, which was being done long ago, but it was banned because of some unfortunate incident But now gene therapy is coming back in a big way. So I will conclude this talk by saying that studying bacteria and phage, I think, is a very fascinating area. When evolution started, bacteria was smiling because the phages could not get in, but they could not multiply here, and therefore the phages are very depressed here. But as evolution proceeds, the phages also have become clear and they have now employed anti-restriction systems, which I never talked about. But I think this is very important. When the phages are smiling, whether the bacteria are now different. So there's always a constant arms race between the bacteria and phage. And at any stage you study bacteria and phage, you never know, you come up with something fascinating. So I want to leave the audience with the thing that doing basic science is very important because you can always use it for an application much, much later on. Exactly what Werner Arber and Daisy Dusswa did. They plated phages, bacteria, and transfected with phages, and look what that resulted. The whole area of biotechnology that has come up in a big way. So I'll stop here and would love to take some questions. Thank you.
Thank you